Hello and welcome to yet another lesson. People in class asked me if I could please continue the videos. So I do. It will also be fine for people at home, right? Okay. So this is a video about 10.4.2. So the point 1 video, if you missed it, check it out on my YouTube channel. And now we're going to continue this piece about what you look like. Okay, we switch over to this screen. And yet it is another very difficult story here, but I'm going to simplify it for you. Genes are shown here. You know that DNA is curled up into a chromatid or a chromosome, and the curling up inside there are the bases of DNA, the 4 letters, A, C, T, and G. And here it says a DNA with a nucleotide sequence of CTAA, ATC, GGT, right? It doesn't tell me anything, but this is a recipe. And it is a recipe for a flower. And the recipe is for what color the flower is. Well, there are 2 types of this recipe. There's the recipe CTAAA and something and something and something. And there is the recipe, as shown here, of ATAA. Notice there's only 1 letter of difference, right? So these both code for the color of the flower, but they both code for a different color of the flower. Right, so genes are like a grocery list. And on a grocery list go things like flour or sugar, eggs, salt, lime zest, and butter. And if you combine those groceries, you can bake a cake, right? And first you're gonna make the dough. So combine all this and mix it all together. You get a dough, and then you put it in the oven, and you get a cake. Well, actually, this is the same. So if you have DNA is the recipe, so the grocery list, and on that recipe is a recipe for a protein. And this protein is going to carry out certain functions in the human body. And those functions you can see outside. For example, there is DNA in my body, there is a gene in my body which tells me what shape my earlobes are. And in my case, I've got the trait, the allele, that they are loose from the rest of my ear. So the loose earlobes are then showed on the outside of my body. Okay, so a very famous organism for showing genetics is the pea plant, because Gregor Mendel, he was a monk, he lived hundreds of years ago. He came up with this idea, and he looked at different traits of the pea plant. He looked at flower color, at seed color, seed shape, pot color, flower position, stem length, and he did all kinds of experiments with that. More about that in the 3rd year. What we're going to do is we're going to focus on a drunken baker, and I tell you why. So these recipes, right, they code, in our case, for a cake. Okay, and there's also other recipes which code for a hot meal and ones which code for a cocktail. So if you combine all those recipes, you got a feast meal, right? You got cakes, you got meals, you got cocktails, so it's a whole party. Well, in making babies, there's also a lot of recipes that work together to form a baby. And remember that a gene is the grocery list, and an allele is the variant of that grocery list. So, for example, you have a recipe for a cake, right? But the allele can be, hey, is it a vanilla cake or is it a chocolate cake? So, for the same for humans. Is the skin of the human vanilla colored or is it chocolate colored, right? Or loose earlobe, attached earlobes, anything you like. Okay, so let's move over. So if babies are mixes of mom and dad's kitchen recipes, then there's a lot of things that have to go right to form that perfect baby. So sometimes things go wrong, actually. And things that could go wrong, I'm making again the link to the baker. Let's say the baker is drunk, or stoned, or ill, when he's baking a cake. Well, the cake then that it's going to make is misformed, right? So let's say mom is ill from pathogens, so she is infected with bacteria or viruses or with a parasite. Maybe she uses drugs during her pregnancy, or she drinks a lot of alcohol. And then the alcohol, the drugs, and the pathogens are also transferred to the baby, and they do damage to the forming of the baby. So the baby will be disformed like the cake. In most cases, babies will have brain damage then. So you don't want that. So that's why we say moms can never drink alcohol or do drugs during their pregnancies. And they have to watch their health, of course. Okay, something else could happen to the baker as well. The baker could, in creating his meal, he could make one recipe twice. So let's say it takes recipe 21 twice. And yes, it is especially this 21. Maybe you've heard of it. There are people in the world who have not 46 chromosomes, but 47 chromosomes in their cells. They have 3 times chromosome 21. If you have that, you have the Down syndrome. And people with Down syndrome, they have 3 times chromosome 21, and the rest of them is normal. So the traits which are on those chromosomes, so the recipes which are on those chromosomes, are expressed twice. They have a very typical face. Most of the time, they're very stubborn people, and they're very happy people, as you see in the picture with their adorable faces. Okay, so what else could go wrong? Well, in copying the recipe, maybe the baker makes a mistake so that he doesn't copy the right amount. Let's say he uses, instead of 200 grams butter, he only uses 20 grams of butter. If you only use 20 grams instead of 200, you get a really, really dry cake. And this really dry cake does not taste so well. Could this happen in human babies as well? Yes, it could happen. For example, if one of the alleles is expressed in the wrong way, the sequence for that recipe, so the way this recipe is read in the wrong order and processed wrong. Therefore, genetic disorders may develop, like cystic fibrosis. And cystic fibrosis is a really bad one. I'm going to show you in class something what happens to your lungs and what happens to the mucus production in your body. Okay, so for doctors, they want to find out very soon if something is wrong with the baby, right? Maybe they can already arrange something to help the baby before it's born. There are 2 types of research doctors could do. This one you're seeing right here is called amniocentesis. And amniocentesis, there they punch a little hole inside the placenta and inside the amnion and the chorion, and they take out some amniotic fluid. Oh, excuse me, I said something wrong. It's not in the placenta, it's in the amnion and the chorion. They take out some of the amniotic fluid, and inside the amniotic fluid is some DNA, and they can read the recipe of the DNA and see if it's still right. Sometimes, however, they need to do some more research, and they do this research. This research is called chorionic sampling. And chorionic sampling means that they do take a sample of the placenta. Sometimes that's needed to have a little of the tissue over there. And the way they do it, you see it here. They go in with a catheter and take a little of the chorionic villi out to do some research about. All right, so to sum it all up, we have recipes, and those recipes are genes, and those recipes, they combine to something like a cake, and the cake is a part of the meal. So in a whole meal you have cakes, and you have meals, and you have cocktails and whatever. So in real life, in the process of baby making, there is the combinations of all those genes, all those recipes which form the baby with all of its traits. And all the traits you can see on the outside are called the phenotype. So, and all the recipes which are inside the baby, you call the genotype. So there's phenotype, what you see on the outside, so the end product, and there's genotype, which is on the inside, to show what the recipe actually is. Okay, I hope it helped you. And now you'll be eligible to fill out the rest of the exercises for 10.4. Okay, I wish you good luck, and I hope to see you soon in class. Bye-bye.